Hello, my name is Kimberly Woodzanowski, and I am a fourth year graduate student working in the lab of Dr. Catherine Grimes. I am honored today to tell you about our lab's research on chemical glycobiology and innate immunity. The Grimes lab is generally interested in understanding how mammalian cells recognize and respond to the presence of a pathogen. We take a chemical biology approach to investigate this problem, utilizing a host of tools such as organic synthesis, molecular biology, biochemistry, and bacterial cell engineering. Our lab has four main research questions surrounding this idea. We are interested in understanding how are the immunostimulatory molecules of peptidoglycan generated? Where are the peptidoglycan immunostimulatory molecules generated? How do cell wall fragments and the innate immune receptors interact? And what is the mechanism of receptor activation? One of our projects to begin answering some of these questions is studying the leucine rich repeat or LRR domain interactions with bacterial peptidoglycan. Candida albicans is the most common pathogenic fungus found in humans, and though it is normally commensal, a morphological switch to filamentous hyphae allows the fungi to cause infections. Hyphae formation is controlled by the adenyl cyclase CYR1. The leucine rich repeat, or LRR, was found to bind bacterial derived carbohydrates, shown here above, with high affinity and elicit this hyphae formation as shown on the left. Our lab also uses surface plasmon resonance, or SPR, to study the interactions of LRRs with the peptidoglycan derived fragments. SPR is an analytical technique used in our lab in which small molecules are tethered to a self-assembled monolayer, shown here, to measure the binding affinities for peptidoglycan-derived fragments to these NOD-like receptor proteins. Small molecules that are tethered to the monolayer are synthesized, such as the 6-amino monomer dipeptide fragment shown on the right. Using this technique, we have determined binding affinities of NOD2, NALP1, CYR1 proteins for PG fragments. We also use this technique to gain insight into the binding pockets of these proteins by synthetically designing molecules that attach to the monolayer at different positions. Our lab is currently utilizing cross-linking probes along with purified proteins to identify the binding site for these peptidoglycan protein interactions. Another one of our projects nicely pairs carbohydrate synthesis with bacteria engineering to study bacteria's cell wall growth. Our lab has developed multiple syntheses, including the CBZ protecting group root here, to synthesize 2-amino-NAM. Through bacterial engineering, we can incorporate recycling enzymes into different species of bacteria and get them to selectively take up our probes. Then we use click chemistry to fluorescently label the bacteria cell wall, which can be visualized using super resolution microscopy. We have also developed 3D synthetic intestinal mimics and hope to use all of these tools to pull out and identify naturally produced bacterial fragments from an immune response. We are also interested in studying receptors within immune cells that interact with pieces of bacteria when they are broken down in an immune response. NOD1 and NOD2 are cytosolic innate immune receptors responsible for recognition of bacterial derived carbohydrate and peptide fragments of the bacteria peptidoglycan. Molecular recognition of these fragments elicits a downstream signaling cascade to generate an immune response. This process is regulated and influenced by dynamic interactions of these receptors with different protein partners, such as HSP70. Besides, NOD2 and NOD1 degradation, which has never been addressed before, is also of our interest. Understanding molecular level details about the interactions and subsequent downstream signaling of these NOD-like receptor proteins by peptidoglycan is critical in the development of targeted anti-inflammatory therapeutics for NOD1 and NOD2. In addition to studying protein partner interactions, we are interested in studying how these proteins may be modified with different carbohydrates. O-glycinylation is a post-translational modification in which O-glycinac transferase, or OGT, transfers N-acetylglucosamine, or glycnac, from the UDP glycnac to selected serine and threonine residues, and O-glycinylase, or OGA, removes O-glycinac of the target. CETSA is a cellular thermal shift assay that our lab has recently shown is a convenient method that can be used to detect the stability effect of olignacolation on NOD2 and NOD1. Lastly, we have combined synthesis and biochemistry again to understand the environmental dependence of NOD-like receptor binding. NOD2 is known to be membrane associated. Using a combination of chemical synthesis, analytical assays, and protein chemistry, we tested the binding of synthetic peptidoglycan fragments to NOD2 in a native membrane environment. Only when the protein was presented in the native membrane did the binding measurements correlate with a NOD2 dependent NF-kappa B response, indicating that this membrane confers ligand specificity to NOD2. Our current research takes these peptidoglycan probes, as well as other more complex disaccharide fragments, and assays them for differential gene expression using RT-PCR and RNA-seq techniques. Overall, our lab incorporates a wide variety of techniques, such as synthetic organic chemistry, chemoenzymatic synthesis, cloning and protein expression, protein purification, cellular imaging and microscopy, mammalian tissue culture, biological activity assays, cell engineering, biochemistry of innate immune receptors, and protein and small molecule NMR to answer questions related to chemical glycobiology and our innate immune system. Our work would not be possible without generous funding our lab receives from a wide variety of sources shown here. Thank you for listening about the diverse work going on in the Grimeslab. If you have any questions about any of our current projects, feel free to reach out via email or read some of our publications on our group website. We hope to welcome you into our team this fall.